So welcome to everybody. It's really a pleasure to be here today. First, I would like to thank the ERN committee for giving me the opportunity to present this talk, and I would like to present my colleague, Professor Martin Turner, who is Professor of Clinical Neurology and Neuroscience within Oxford University and honorary consultant neurologist to the John Radcliffe Hospital in Oxford. His research focus over the 20 years has been the development of human biomarkers, including advanced neuroimaging and biofluid analysis, to accelerate therapy discovery in ALS. And Martin, it's a pleasure to hear you presenting a Bayesian biomarker-led drug screening platform in ALS.
Great. Well, thank you very much, Philippe. And thank you very much to the organization for inviting me to do this, and Vincenzo for the invitation. So yeah, I'm going to talk about and hopefully what you'll think is an exciting new development in the way that we're starting to think about developing therapy for ALS. Just to get everyone clued in again, I mean, you know, the original descriptions of ALS by Charcot, the very classical syndrome, hasn't changed, of course, and you know, it's very much a clinical diagnosis. This combination of lower motor neuron features you can see marked in green there, and the upper motor neuron features that we mark in red on this, and that combination of pathology is really quite unique in spanning both the sort of central and peripheral nervous systems. But we recognized in the last 20 years that there's very clearly a wider motor system involvement, and it's to do with how the motor system connects with the frontotemporal parts of the brain. And some of our patients will develop a really predominant frontotemporal dementia, and there's a spectrum of involvement across that. So it's become much more of a system disorder, but still very much characterized by those key clinical findings. The symptoms you'll recognize come largely the disability from the lower motor neuron involvement, leading to a lot of muscle atrophy and weakness. The stiffness of the central nervous involvement is disabling, but to a lesser extent. But it can create problems in combination that make us need to address swallowing issues, nutrition, and ultimately that's going to start to affect the diaphragm, and that leads to the sort of median survival that we unfortunately recognize with ALS, which is only 30 months from the first symptom onset. And we also increasingly have to deal with cognition and behavioral symptoms, even in patients who don't develop frank frontotemporal dementia. So just thinking about, before we get into sort of ideas on therapy, what's the cause of ALS? Well, there isn't one cause, and it's multiple causes really. And it's kind of like a large downstream river that's fed by lots of upstream tributaries. Most of the disease passes through this pathology, this protein pathology, I guess our version of amyloid that we call TDP-43, a 43 kilodalton RNA binding protein. And that molecule defines the disease to a large extent. And most of the cases, whether they are so-called sporadic, they'll pass through that on this diagram, and they end up mostly as ALS, but as I've mentioned, some cases of FTD, and there are other genes that just lead to FTD, such as MAPT and progranulin. But we also recognize, if you look at the bottom part of that, there are a couple of genes, SOD1 and FUS, where they lead to the same ALS sort of phenotype clinically, but they seem to spare this TDP-43 pathology. So it's a complex situation, and the gene that we recognize is doing most of the work really, certainly in Western European populations, is this expansion in chromosome 9, open reading frame 72, which is a large hexanucleotide expansion. In terms of mechanisms, I guess, you know, we often call ALS motor neuron disease in the UK. It's a sort of historical term from the 20th century, but it's it's probably a bad name all round, really, because it's very clear that it's not just a motor problem. It's certainly not just neurons involved, and it's certainly not a single disease. And if we look at this interplay of the blue motor neuron here with its surrounding astrocyte and microglial interactions and also interneuronal interactions, we start to realize there's a huge number of pathways we can find that are damaged. We can find problems with axonal transport. Motor neurons are undoubtedly extremely long neurons and have um, a requirement to transport materials and nutrients a long way from the cell body out to the muscles. We've long thought that there's been evidence of mitochondrial dysfunction. Too much excitability or an imbalance in excitation and inhibition has been a long-standing mechanism, and that's thought to be how the drug riluzole might be helping slow the disease to a small extent. Oxidative stress still remains a very strong candidate for a long-term loss of protein homeostasis. Neuroinflammation is an increasingly important area. We recognize that microglia come in to clear up the damage, but undoubtedly they may be put into a state where they end up causing more damage. Although whether that's a primary event, I think, is more controversial. Interneurons have always interested me for at least 20 years, and I still think that they're a strong candidate really for being a major effector in the process, simply because of their sort of position in the circuit, both centrally and peripherally. And so there's a lot of interest there. Astrocytes, I think, are now increasingly being implicated as potentially involved. And you can start to see that it may not be a primary problem with the motor neuron. It may be a problem with support cells. RNA processing is a growing theme. Nucleus transport, apoptosis, and this sort of cellular biophysics that we call phase separation. So realization that the complexity of protein synthesis and transport within the cell requires almost at a quantum level, really, changes in cellular physics that we're only really starting to scratch the surface of and understand. And one of the sort of more recent mechanisms is the ability for what are normally cryptic exons or hidden, not expressed exons are released by the loss of TDP43 control, and so they might be again involved in initiating disease or promoting disease. So you can see that there's a huge number of pathways here, and this is both exciting but also a big problem because it makes it very difficult to rule things out when we get results from all sorts of biomarker studies. It's very hard to find a result where we would not be able to fit it into any of those pathways. And it makes us increasingly want to understand what the hierarchy here is among all this sort of possibility. Now, if you go back to the patients, well, we have most of our patients falling into a prognostic track. This is a survival probability curves across a large number of patients. That was a large European collaborative project. And you can see that most people fall into the Lou Gehrig sort of model. So Lou Gehrig, a baseball player from the 1930s who gave his name to the disease in the US. And he had a very typical progression, median survival, 2 to 3 years. And that's at least half the patients. And of course, we have exceptional cases like Stephen Hawking, whose disease course was 50 years. I think the important thing to say is that individuals seem to set their own pace. And so when the disease is slow, it tends to remain slow. And when it's fast, unfortunately, it doesn't tend to slow down. And among all these different rates of progression, and what we've done here is just put most patients into one of sort of 5 categories. All of them, nearly all of them, bar those rare SOD1 and FUS cases, will have this TDP43. So in all this variability, we have a unified pathological marker. And what that means is that it is possible to slow this disease down. There is obviously, if we take the view that if they have the same pathology, they are the same disease, and yet we can see that there is a variation in the speed, that tells me that this is not an impossible disease to cure. This is not a condition which we have no possibility of slowing down, because there are biological reasons we just simply haven't discovered yet as to why people move into are in different curves. And so that gives me great hope that it is possible with the right drugs to shift the curve for an individual and to slow the disease down. So in order to kind of give someone a drug, one of the problems with ALS in a way is that, often strikes me as amazing, really, that our patients with ALS, despite often a rapid deterioration, they don't feel unwell. They don't feel systemically ill. They obviously feel weaker, and they may have some secondary joint pains and discomfort. But it's a disease which is happening really in plain sight. The immune system isn't super activated, and the blood tests are all normal. Often the patients have been told all their tests are normal. Um, and so it's a condition really where if we gave someone a drug, we can't expect them to say, "Oh, I feel better," because they didn't feel unwell in the first place. And so it becomes a matter of how are we going to know if we give a drug that it's doing some good? And of course, we're not naive enough to think that we could give a drug and someone would feel better the next day. Um, but we have to kind of get better at measuring disease activity and how disease progresses. Now, that would allow us to get people into trials earlier. If we can make the diagnosis sooner, that's a complicated issue. It's not really a failure of neurologists to make the diagnosis earlier. It's how it presents, really, this insidious presentation, often in very fit and athletic people who haven't been ill before, and so they don't assume they've got some life-limiting disorder like ALS. We do want to stratify patients so that we kind of have a sense of who's in a fast and a slow track. So that if we do a trial, we're comparing like with like, and we're not expecting someone with a very fast disease to necessarily respond in the same way as someone with a much slower disease. And within that, there are regional variations, patients who start with their upper limb, lower limb, bulbar involvement. So we want to kind of understand biomarkers that reflect that. But the key thing is this biomarker really that's sensitive to changes in disease activity that would allow us to give a drug and then perhaps be able to say, no, it's not working, let's stop and move on. And many of our patients will try all sorts of things that they get off the internet, various supplements and vitamins. And they want these biomarkers so that they can have some sense of whether what they're taking is making a difference. Now, I'm very lucky in Oxford that we have the full kind of what I call neuron to network portfolio of biomarkers. So we can go from the extremely small, the DNA and cell biology, which my colleague and head of department, Professor Kevin Talbot, has expertise in, through the biofluids into brain imaging, and then the wearables. And my colleague Alex Thompson in the middle there is very involved in a number of those aspects. I've tended to focus on the imaging over many years, and then in the last 10 years, link that up much more with biofluid analysis. Now, how do we measure what's happening in a trial, really? How do we do it? Well, we've got two options, and the one on the left, option one, is to do a survival study. So we take a bunch of patients, we give them drug X, and we give the other half a placebo. And we look at the difference after 18 months, and that's a long time. For a patient with a median survival of 30 months, that is a very long period to have to study them. And of course, half of those patients will be on a placebo. Option two is slightly shorter, but is messy, because what we're using here is something called the ALS Functional Rating Score. You can see that sort of set of lines that looks like a bird's nest. They're showing the score falling over time as the patients get more disabled. And some of those scores fall very quickly, and others fall much more slowly. And if we put them all together and sort of average them, then we get those thick red lines, and we say, okay, we want the people on placebo, their rate will likely stay the same. And the people on drug X, their rate may slow down. So we'll get separation of those curves. And that's very, very messy. it takes at least 12 months. People try hard to do that in a shorter period of time. We're all pretty sure that 6 months just is not long enough to get that answer, and 9 months probably isn't long enough either. So again, 12 months. Now, this means that, to power these kind of studies, you need about 300 participants minimum. You're going to have to recruit those participants over 12 months because not everyone starts the study on the same day. And it's going to be a multi-site study. That's going to take a lot of organization, bureaucracy, all the ethical approvals, and it's going to take 4 to 5 years to do those kind of studies from the start of the idea to the final result. And half of our patients, at least, won't survive that trial to even get that result. So this is a lot that we're asking of patients, and I think we've done this for 30 years. We've had lots of drug ideas and we've tried them, and it's becoming unacceptable really to do this without greater sort of certainty that what we're doing is the right thing. We've used over 100 different types of drug. I've sort of grouped them here in categories. This has involved, I estimate, 25,000 patients over the last 20, 30 years. You can see the ones in capitals are the large multi-center studies, all of which were negative. Riluzole was a positive study, but sadly doesn't work to the level that we'd like it to. You can see I put interleukin-2 there in sort of orange. We're waiting to see if we can get a phase 3 study off the ground. There were some very interesting signals in the phase 2 study published in the Lancet Neurology, but it needs a big study, and so the jury's out on that one really. There's no shortcut to doing the definitive phase 3s. So lots and lots of studies, lots and lots of patients, time, effort from them. That's the main thing, but also the cost involved that we all have to think about because we have to fund the staff to do these. So, the good news is we've got a huge number of drugs that are waiting to be tested, but there is simply not enough money in the world really that we could throw at this because to get all of those tested in a phase 3, there aren't enough patients to do those studies, all those studies, and there aren't enough staff to run those trials. So we simply can't rely on a system where it's whichever drug company has the most money or shouts the loudest. That leads to one or two of these type of phase 3 studies every year. As I say, the track record is very poor and they take a long time. So just a reminder that the key obstacle that we've got, I think, to moving forward and doing this faster is measuring disease activity. Something that you could measure that would say, okay, this patient's disease rate, their kind of speedometer on a car, their speedometer is showing this, and therefore if we give a drug and we can slow that, then we're making a difference. So the idea that I'm going to sort of pitch to you really is that there's a third way to do things, and what it involves is trying to get a human signal. So a signal that's not from a mouse or a dish, but from a patient, but it gives you a hint that you're moving in the right direction. That doesn't mean you can then just do it in 3 months, but it gives you much more confidence then. And this is something called experimental medicine. It means that you're using the human model really to test a drug, to get a readout, a surrogate of disease activity, which you then can put that drug, if it's effective, you can put that drug back in to the more traditional placebo control model. So it's not a shortcut to not having to do a phase 3, but you can imagine if I said to a patient, we're going to do a placebo-controlled trial, so there is a chance that will be on placebo, but we've already done a screen, and this seems to change a surrogate marker that we believe is showing reduction in disease activity. So we've got much more confidence from a human marker that this will work. So where are we going to get these markers from? Well, I'm going to mainly focus, I'm going to focus today on something from the blood, but just to say, I've spent a long time, as many of us have, looking for changes in markers at the brain level, so MRI, and more recently, I'm much more interested in network-based markers using techniques like MEG, magnetoencephalography. People have looked hard at markers at the sort of central pathways, transcranial magnetic stimulation, and of course the downstream muscle EMG-based markers, surface EMG. I think it's fair to say that we've all struggled to find markers that are sensitive. We can find things that are different between people with ALS and healthy people, but that's quite a low bar, really. If you've got a disease like ALS, you would expect to find some differences from a healthy person. The question is whether it's sensitive enough to do more at an individual level or a smaller group level, and that it's sensitive to change. And that's been a big problem over many years of looking for biomarkers. So I want to focus in on this particular blood molecule called neurofilament light chain, which I'm sure many of you will have heard of. Now, this is not new. This has been around. In fact, the first study in ALS was done in 1996, so it's 30 years ago. And it shows you really how things change, because the data are all there if you look up that paper in 1996. But what's changed is the ability to measure this in a confident, reliable way, and particularly in the blood, not in the CSF, which is obviously more difficult to get hold of. Now, it's a molecule that it does have some complexity in its biology, but largely speaking, we think it represents the fact that neurons are degenerating or being lost or damaged. So it's a part of the makeup of neurons, and is released in response to damage into the CSF and then ultimately into the blood. Now, a really key message about neurofilament light chain is that the level equates to a rate of the process that's happening. So it is a speedometer. So lower levels mean the disease is slower generally. So that's why you're seeing on these diseases here, which on the right hand side in the grey boxes, you're seeing pretty low levels of neurofilament light chain, and those diseases there are Alzheimer's, Parkinson's dementia, and Parkinson's disease. And these are very slow diseases. They tend to process over 10 to 20 years, and so they have a low speedometer. They have a low rate of neurofilament. As we move to the other end, the red end, you can see HIV-associated dementia, ALS, and CJD. These are very, very aggressive diseases in a lot of cases. And so the neurofilament level, the speedometer, is generally much higher. And then in the middle, you've got other conditions like MSA, and FTD, you can see, is towards the lower end. So you could almost plot a line through those different diseases that would say the typical survival of those diseases. So on the left, the survival would be pretty short in those diseases, and on the right, it would be much longer. And so the NFL level is equating really to both survival, but generally the rate of progression. Now, in ALS, we know that it's also linked, as we'd expect, to the rate of decline in function, the disability score, and also very strongly with survival. So many groups have reproduced this. There's absolutely no doubt that it's very strongly linked to survival in ALS. So here's how that would look really on those survival curves, that generally, as you move to the right with the slower disease progression rates and longer survival, you're going to find lower levels of neurofilament. And that means that, if you measure neurofilament level and you find that it's low, we would expect that patient to have a slower disease course. And we can already see that in the clinic. We often can predict very early on how someone's doing even before we have neurofilament. But it offers the possibility then that we might just start to think about, could we give a drug and see if we were actually lowering the level of neurofilament as a marker, a surrogate, that we're actually slowing the disease down. Now, a key point about this, and this was, I think, what got me interested in it about 10 years ago, was that I noticed that when we looked at our levels in spinal fluid, that's the blue, green, and red curves there, that each individual line is a patient, each individual dotted line. And you can see that over time, each individual patient stays rather similar. Whatever their level is, it tends to stay the same. And there's some more data there from an actual trial. You can see that on the right, that most of those patients there, their levels stay similar. The individual tracks are marked. There's one where it seems to fall, but all the others really, it seems to be very stable. And then the more spidery plots at the bottom there are two large clinic series. So one that we ran in the UK called the Ambrosia series, and then Michael Benatar's Create group, their data. And overall, this is, I think, the key thing about neurofilament that what the game changer is in ALS is not just that the levels are detectable, because they're certainly usually very raised, but that you know what's likely to happen to an individual, and what's likely to happen to an individual is what we see in the clinic. Their rate of progression stays the same. If they're slow, they're slow. If they're fast, they're fast. And their neurofilament level stays the same at baseline. If you do nothing, it just stays the same. It's not measuring how many neurons they've lost, it's measuring how fast their disease is moving, which tends to be the same. Now, if you know what's going to happen when you don't do anything, then you can start to say, well, what if we give a drug and we were able to reduce the level? Now, do we have any evidence from other diseases? Yes, we do. This is remarkable data on the left, and it's very old now. It's 2007. When we developed treatments for HIV, the highly active retroviral therapy, this study here was looking at neurofilament levels, and you can see that the curves for neurofilament are extremely high when you first start to develop an HIV infection, because you start to get HIV-related neurodegeneration, and as I showed you earlier, some of those levels are very, very high. Then you give highly active retroviral therapy, and within sometimes a few months, the levels have dropped down to normal, because generally normality is associated with undetectable neurofilament light chain. It's not a marker of just simple aging. It's definitely a marker of pathological processes. So you give a highly effective therapy, the virus is suppressed, and neurofilament falls. And actually, there's a lot more data now emerged on the right there. You can see that's just some data from a fingolimod study in MS, again showing that reduced disease activity, both measured clinically and on MRI, was associated with a lowering of the overall neurofilament light chain level. So we do have evidence from other diseases that if you treat them with successful drugs, that's associated, as we'd expect, with a loss and a lowering of that marker, which is a surrogate for the fact that you're slowing the disease down. Now, I think even before these data came out, I was already moving ahead with trying to sort of set up what I'm going to talk to you about in a second, the sort of platform to do this in ALS. But this was the sort of icing on the cake, really, because in 2020, we got the news that this antisense drug for a very rare genetic form of ALS, SOD1 ALS, was starting to slow the disease down on clinical measures. But if you look at that top graph there, you can see that at 28 weeks, there wasn't a significant separation of the two curves, and it actually took a lot longer. And the most recent open label extension data has shown unequivocally that this drug works and actually slows the disease down. And some of the patients, actually in rare cases, have even reported stabilization, occasionally even some mild improvements. So very exciting, very similar to the nusinersen story in SMA. But the key point was that at 28 weeks, that's sort of 6 months, you would not have detected a clinical benefit, which is what I explained earlier. It's not enough time with the measures that we have. But on the bottom graph, look at what happened to neurofilament. That was already taken down really by 50% by the time of 6 months. A massive, dramatic change, showing that the disease process was being switched off, and then the clinical benefit followed, which is exactly what you would expect biologically. So it gives you evidence that an early reduction in neurofilament light chain is what you'd expect to see if you're actually making a disease-modifying effect. And in fact, when this drug was licensed by the FDA, it was a landmark judgment because they referred to neurofilament light chain as a reasonably likely surrogate marker, which sounds a little bit weak, but actually that's very strong language, because it's so far been unheard of for the FDA to accept any sort of biomarker as an outcome measure. So this was groundbreaking. So there's the problem: lots of candidate drugs and very limited access to phase 3. So I think the solution is this experimental medicine approach, and we've called it the experimental medicine route to success, or experts, in ALS. And it's a drug prioritization platform, and that's a key thing. It is not, you know, a way to avoid having to do a phase 3 study, but it's a way of deciding which are the drugs, or which is the drug among all these ones we can test, that we think is the one with the most likely chance of helping in a phase 3 study. Now, I've linked together with my friend and colleague Chris McDermott in the University of Sheffield to do what is a really massive study, and we've divided this up, where Sheffield run the study as the trial unit, and Oxford, we run the laboratory analysis for the NFL. And I'll take you through how we've designed this. It's expensive. It's got funding from what we call the National Institute for Health Research, and they've put £8 million into this 5-year study with multiple centres across the UK. But it's certainly far from the expense of doing a large phase 3 study. And just to give you some perspective there, the last big study, the dexpramipexole study in ALS with 300 patients, multiple sites, cost Biogen $100 million to do that study, which was negative. So yes, these studies are also expensive, but they're nowhere near as expensive. And we've had some generous support from the charities as well, Motor Neuron Disease Association, My Name's Doddy, and also LifeArc. So how it works is we've initially started with repurposed drugs, but they go through a rigorous process. So a panel selects drugs that have been shown in laboratory models, and that's cellular models, rodent models, all of the data is appraised, to have a relevant effect in a pathway of interest. But crucially, it has to have good CNS penetrance. We then randomize patients to one of 3 arms. Everybody gets a drug. It's all open label. We have to make a decision on dosing. And that is a little bit of a limitation. We have to sort of make a decision really on, generally with repurposed drugs, we go for the usual kind of dose that patients would take for the other indication. The way it's designed, we have participants are only going to be in this study for a maximum of 6 months each. So each person is being offered the chance to take a drug, to help us screen a drug, and their involvement will be up to 6 months. And the new design is this, what's called a Bayesian analysis, and I'll talk a little bit more about that. But because of that, we're able to make a decision on each arm with just 30 to 80, and 30 is the minimum, 80 we think is the absolute maximum. Participants per arm to make a decision, with each person only taking that drug for up to 6 months. And the idea is that these are one of 3 outcomes that the modeling, the Bayesian model, will decide. And the first one, you can see at the top in red there, is it detects that the neurofilament levels are starting to rise. That's not good. We don't like the look of that. And if it's consistent, then the model will say, okay, we're calling it, that arm stops. The middle is the more difficult one, because that's the most common outcome, is it's just not doing anything. The patient's carrying on their normal neurofilament level overall. There's a little bit of variation, of course, between individuals, but overall, the mean group neurofilament level is not changing. And that's the one that, of course, requires the most statistical power to be certain. You know, showing that you're certain there's been no change is the one that requires the most statistical power. So that's where the kind of potential for the numbers to be more than 30 arises. But of course, what we really want to see is the bottom one, which is where we get a clear indication, and we've chosen a 30% drop. So not as much as tofersen, not as much as that antisense drug, but a sizable drop, not a trivial sort of 10% drop, 30% drop. And the first 2 trigger us to say, no, that drug's no good, let's move on. and then the last one, that would trigger us to say, this looks good, let's put this one into a phase 3. Now, Bayes' theorem is not new. There's Thomas Bayes from whenever, probably 1800 and something. This is about conditional probability, and it's really what we do in medicine, really. So, when you're examining a patient, you listen to their history, you've got some ideas. You may be expecting them to have a set of central nervous system signs, so you start to examine them, and then you find that they've got some lower motor neuron signs you weren't expecting. So you had a prior probability, and now you've been given some new information, and so you adjust what you now think is going on based on what the new probability is. And that's just that updating is kind of how we use our brains, really, and it's certainly how we do medicine. And it actually allows you really to get to answers more quickly. And it's interesting that it's just this year the FDA has started to realize that this is a very powerful way to do trial analyses, because it allows you to get answers more quickly than simply waiting till the end of the study, and all the data being in, which of course leads to extra time. So I was very lucky. I think what helped this study get off the ground the most was a chance meeting with a couple of statistical modelers, both of whom had come over from Ontario in Canada, where they've been very heavily involved in some of the COVID modeling. And Peter Uni has a background in cardiovascular and cancer therapy. And Peter chatted with me, and I said to him, this is what I want to do. I'd like to have a small group of patients and be able to give them a drug just for a maximum of 6 months, and then to say, is it changing their neurofilament level? And I gave him the data from the CREATE consortium, and also from the Tofferson published study, and also from our Ambrosia study in the UK. And he put all of the data, the natural history of neurofilament, into his model. And he came back to me and he said, oh yeah, I don't know anything about ALS. I don't know anything much about neurofilament, but he said, this is a superb marker. He said, we would love to have this in cardiovascular disease and cancer disease. For what you want to do, this is perfect. It will give you a reliable readout as to whether it's not changing or going up, they're sort of futile outcomes, or whether it's going down significantly. And by using a Bayesian model, you can do interval analyses and make the call as you go along. So when the model feels it's got enough data, it can make the call and say, right, we're stopping that arm, or okay, it's working, and then we stop and we put it into a phase 3. Now, on the flip side, we want to obviously pick the right patients, and we've been very working very closely with Ruben van Eyck, who is well known in our field for being very interested in trial design, and he's developed a prognostic tool that we can use called the TRICALS prognostic score, or risk score, which is just some basic clinical measures that ensure that we get the balance right between giving as many people access to this study as we can, and we think we can probably admit about 75% of ALS patients into this kind of study, but making sure that we don't put patients in whose disease is fortunately for them too slow, but for the model there wouldn't be enough change really to power the model. It's much less restrictive for people who have a rapid prognosis, provided we get them early, they can go in. But I think it's a universal problem in trials that it's just very difficult to include patients whose disease is very slow. That doesn't mean they wouldn't benefit from the drug in the longer term, but I think for the purposes of the trial, we have to make sure we get people who are going to actually power the model. Now, the first 3 drugs that we're trying, we've been running for about a year now, and this is a brand new 10-site trial. So recruitment's always a bit more challenging than you expect, but we're kind of now up to the sort of numbers where we're starting to contemplate our first interim analysis, and that could be very exciting. Whatever the results are, if the interim analysis says, look, we've recruited 30 patients to each arm, and it's very clear that it's futile, that the levels aren't changing with any of these drug arms. Yeah, it's disappointing in a way, but it means that we've just excluded 3 drugs that would normally have taken 4 to 5 years each to have, and 300 patients per trial with placebo to have ruled out. Now, all 3 of these you will recognize with their original kind of indications, but I can tell you that in laboratory they have dramatic and remarkable effects on motor neuron survival. So metformin, rapinol and nifedipine are all extremely plausible repurposed drugs. And we've got at least another 6 lined up ready to replace these if we make the futility call. Now, unsurprisingly, industry has started to sit up and say, well, hold on a minute, we could save millions of dollars here if we've got a drug that is often the case. They're just interested in trying in a number of diseases. That's fine. I understand that. And they're often interested in ALS because they can get an answer reasonably quickly. If they can save money and get a signal through experts at ALS, that's a huge bonus. And one company that have now released a press release, so I can share with you, is Raya Therapeutic, who have a compound which is a neurotrophic stimulator. They're very interested, and we're working towards bringing that novel drug to experts. So the idea is really that with time, we'll start to move away from repurposed drugs and into novel compounds that have obviously been tested first for safety in all the usual ways. This is to show you how this study works. We have an NFL level that is done 2 weeks before baseline, and then the baseline is when the patient's randomized and starts taking the drug. And that marker or that measurement either side of that is just a way to calibrate the model, because of course there's a little bit of variability within an individual for neurofilament, and it will certainly detect if there is an unusual spike. Obviously things can happen, patients can have a knock to the head or something, and so we have to calibrate the model so it can weight that appropriately if the marker shows a big difference. We then take another blood sample at 12 weeks, another one at 18 weeks, and then the last one at 24 weeks. Now, those last ones, 18 and 24, are critical really to the modeling. And then we carry on with consent. We just do a sort of natural history data collection out to a year, but the study itself stops at 6 months for each individual. Now, running alongside this is a very, very exciting biomarker development program, because you can imagine we're collecting a lot of samples here, and within each group you'd expect individuals to do different things. It's always at a group level that we're measuring the neurofilament, so we want to know if there are certain people who are showing a different trend, and to understand perhaps why people respond and other people don't respond, and to look at that across the whole range of omics, and actually all the way out through to things like surface EMG. And what we're doing is looking at MEG network changes. So that's a kind of biomarker program that runs alongside. So coming to the end, what are the questions this raises? And obviously we may come back to these in the question session, but people often say, well, hold on a minute, there might be some really important drugs out there that can help improve disease activity and improve survival, but they don't affect neurofilament levels. Well, I don't buy it myself. None of us should have raised neurofilament levels. It's not a normal marker. So a healthy state is to have low neurofilament. So if you're making the case that there's a remarkable drug that is fixing ALS, and yet apparently the patient's NFL level doesn't change, I don't believe it. So I'm not worried about that. I don't think that this is a spurious marker. It's possible if we lowered NFL that we didn't see a clinical benefit. That might be because we have to wait longer. But I think, that again, we've got evidence from other diseases and from tofersen that that's not the case, that there is a very tight relationship. It's possible that wanting to lower 30% is quite a high bar, but I think it's a reasonable place to start, because we don't want small effects. People often say, what does riluzole do to NFL? Well, I don't know, but I suspect not very much really, because it doesn't seem to slow the disease down very much. So we're not interested in 10, even maybe 20% reduction in NFL. It's a bit too noisy, really. We want a big reduction. Obviously, in HIV, we saw a complete reduction in tofersen antisense therapy, 50% reduction. We want to make sure if we're seeing a signal that we believe it. So I think 30% is a reasonable compromise. There is, I think, a definite concern that there are some drugs that might cause a rise in NFL initially, perhaps ones that affect microglial function. And what we don't want to do is throw them out early on. And so the model is designed not to kind of make an immediate call if we're seeing a little bit of a rise in neurofilament, only if it's a sustained rise. And many of you will be aware that the Huntington's drug that was tried in the past, not the more recent one, but in the past, one of the reasons for stopping that study was a sustained rise in NFL. And indeed, I think that the consensus view that was a warning sign. So overall, I think that we should be alert to a rise in NFL. That isn't something we should ignore. It's possible that we might put some people into a study whose neurofilament level is just still rising before it kind of plateaus, really. That's unusual, though, to get someone into a drug trial before about 6 months of symptoms. So I think that's not a huge concern, actually. There's definitely issues around the assay platforms. It's taken us a huge amount of work to get the platform. We're using the Simoa platform just because it's well tried and tested. It's sensitive, but it has batch effects. We have to batch the samples. We have to correct across different lots of different batch lots of the assay. It's very complicated, and certainly in the UK, we have to be working at what's called good laboratory clinical practice standards, which is almost as high as a hospital laboratory standard. In order to make decisions about starting and stopping arms, our regulatory authorities require us to do quality assurance on the assay, and that is a hugely time-consuming aspect. Now, I'm hoping that with time, we'll develop much more reliable studies that you can do at the bedside. Will industry bring their drugs? Yes, they will, and they already are. So, some people are a little bit skeptical about neurofilament. It's not perfect, but I think it's the single biggest gift that our field of ALS, after years of failure, it's the biggest gift we've had, and it is very good. It's not perfect, but it's very good. I think that just trickle of studies into phase 3 is not good enough. And, let's remember that people living with ALS, they'll get one shot at a phase 3 in their lifetime. So it is up to us to make sure that we're giving them the best odds of success in that trial. And one of the ways to do that is that we can say to them, look, the drug we're trying has been through a screen and it lowers this neurofilament. That would be a massive difference. So I'm changing the conversation in clinic. I'm saying that it's not acceptable to carry on doing trials in the same way. Humans are the best place to look for surrogate markers. It will improve the odds of success. It's not experimental medicine in the sense we're experimenting on patients. That's an important, message to get across. These are drugs that, even the novel drugs that have been tested for safety, but experimental medicine means we're using human responses to choose the right drugs. We don't know the key pathway target in ALS, so we shouldn't be too reliant on saying, oh, this drug's the most important because it's doing this. Repurposed drugs do have value, and I would not be at all surprised if any of the repurposed drugs that we've selected turns out to have some benefit. And certainly the history of medicine has several examples of where that's been the case. Industry, I think, are getting more and more interested. And so what we're offering is to patients, we're saying, look, there's a choice here to help us in drug discovery. And many patients are very keen to do things which are for the greater good. It's not something we can take for granted. But my experience of 20 years of working with patients is they understand that they have to help, the group effort, really, that's the only way we're going to get to the answers. So I'm saying, look, everyone at diagnosis gets offered riluzole. That's pretty standard. And then they should get offered participation in experts. And that's going to take them out to 4 to 7 months from diagnosis. And at that stage, they can then still have the choice if they want to go into a phase 3 study, because, as I said, the reality is that will probably be their one chance of a phase 3 study. So experts is not taking people away from those kind of studies, but it's allowing us to get some candidate drugs much more quickly. So last slide is just to show the huge team involved in this. This is easily the biggest project I've ever been involved in. It's a massive collaboration with Sheffield, and I'm enormously grateful to my colleague Chris McDermott. We work very closely together on this. Huge team of people at the Sheffield Trials Unit, and then the people running the lab assay on the right there. Amar Al-Chalabi, Pamela Shaw, and Kevin Talbot overseeing the drug selection choice and the modelling I've shown you there, and our expert patients in the middle who are always guiding what we do, and making sure it's acceptable to patients. So thanks for your attention, and I'm very happy to take some questions.
Thank you very much, Martin, for the comprehensive and very instructive talk. I have some questions for you, of course. And the first one, is it for you the neurofilament? Is it for you the best biomarker we can have currently? Or do
Yeah,
you
I think
think?
so. I think so. I think it's easy to measure. The measurements are reliable. They're getting faster. I do think that it will only be a matter of a year or two. We'll be doing these tests, um, on actually at the point of clinic. We'll be doing them like a kind of full blood count. So it's it's a marker that's predictable. It's stable. The samples can be left lying around and it doesn't degrade. It's great, for what we need it for. It's not very good for other diseases like Alzheimer's and Parkinson's because it's just too low. But for what we need it for in ALS, I think it's a wonderful development, and we'd be very foolish to pass it over.
And my second question, what is your position concerning troponin? Is it for you a future for evaluation of drug efficacy or
Which drug was that? Sorry.
troponins? The troponin
Oh yeah,
2.
troponins. Yeah, well, some very interesting data on those. I mean, I think that they're a little less sensitive, compared to neurofilament. So, but the idea of combinatory biomarkers, I think, is definitely
Mm.
attractive. Um, but I think on the current evidence, they're not as sensitive, as SOD, or troponin I specifically is not as sensitive as neurofilament.
So if I understood the literature for me, some publication suggests that neurofilament is a good marker for CNS and hypomotor neuron involvement, and troponin a good biomarker for low motor neuron involvement. Do you agree that?
Yeah, I've seen those data. I don't think it's quite clear. Initially, we thought it was a lot of, because of course you're finding it in the central nervous system when we do CSF, the neurofilament light chain. But I don't think it's quite that clear cut. We do see raised levels in
Mm.
SMA. So, but I think the troponin probably is a bit more reliably, a lower motor neuron marker.
Yeah,
Um, so yeah,
I agree.
it's more about the sort of sensitivity to change, really. I think that's the key.
Okay, thank you. We have two questions from the chat. The first one is from MM. Since ALS therapies target many different biological mechanisms, some of which may not directly reduce neuronal injury, how does the expert ALS platform ensure that potentially beneficial drugs are not overlooked if they do not produce clear changes in biomarkers such as neurofilaments?
Yeah, I suppose one example is, we tested a drug as in a multi-site way in the past, tirasemtiv, which was a neuromuscular potentiator. So it was sort of
Mm.
increasing downstream neuromuscular transmission. Um, so perhaps improving, measurements such as forced vital capacity. I could imagine that might not affect the neurofilament levels, yet still, has some benefit for patients. But I think it's hard to imagine, as I say, none of us should be walking around with raised neurofilament levels, significantly raised levels. There's something going on in the nervous system if your neurofilament levels are raised, and therefore, if you're not changing the disease at its core and stopping the neuronal loss if you're not lowering the neurofilament. So I think if it's a truly disease modifying drug, it must affect the neurofilament levels. It must lower it. So I'm not, I wouldn't be worried about throwing out a drug that's not changing neurofilament. I think the chances of that being an important drug for ALS is low.
Okay, but if I'm right, riluzole has no effect on neurofilament.
Well, the studies haven't been huge, but people have tried to look at that. But yeah, I mean, I just think it would be astonishing, really. It would almost
Yeah,
be if
yeah.
it had a dramatic effect on lowering neurofilament, I would then be pretty depressed, really, because I'd say, well, it's not what I see in the clinic. I don't see the patients, tangibly slowing down. We know in the trial they do, because of course you did the trial for 18 months, large numbers,
Mhm.
and you can see small effects. But
Yeah.
we don't want any more small effects. We've had enough 20, 30 years of small effects. We want a big effect. And that's where
Yes.
neurofilament comes in.
Yes. Vincenzo wants to intervene. Yes, Vincenzo.
Yeah, Martin, excellent talk. I have one main question could be, what about riluzole? Is riluzole decreasing, increasing neurofilament in your experience?
Yeah, well, we were just saying that I think there's not really any evidence that it does, but that doesn't surprise me because it's got such a small effect. So I think, yeah, I'm not losing any sleep over riluzole. I'm after something that really is making a big difference for patients, and I think if we want to make a big difference, it's got to lower the neurofilament convincingly.
And when we talk about neurofilament, your goal is in the fluids, in the CSF, or in the blood?
In the blood.
In the
Blood.
blood.
Yeah, it's a very tight. If you plot CSF levels against blood levels, it's extremely tight. I mean, almost 0.99 correlation. I mean, it's extremely tight relationship. So that's one of the huge advantages, and it's very stable as well. So although we do make sure the samples are collected in a very rigorous way for a trial purpose, the reality is, because we've done the studies, you can leave it lying around on the bench for a week. It makes no difference to the marker. It's extremely stable neurofilament, so it's very reliable.
And the last question is, I'm always concerned about the biology. I don't think we don't know anything about the biology of neurofilament. I'm really, the conundrum is why is stable in the same patients? I mean, what?
Yeah. It does puzzle people why the levels stay the same. I mean, they clearly do. As I showed you the natural history data, it doesn't seem to drop off. Now, I suppose we should just remember that you're not, no one's going to ever run out of neurons. So when we all die, I'm sure we've still probably got 90% of our neurons. So we're never going to see it dropping off because we've run out of neurons to generate. So I think it just simply reflects the equilibrium of loss and transport, because it will get absorbed, taken up by other cells. So I think it's just reflecting the individual's equilibrium and the aggressiveness of their disease. It's certainly not, the level is not a kind of reflection of the actual number of neurons that have been lost.
So it's basically from neuron in your perception.
I think
Mm.
so. I don't think we
Okay.
can be too pure about that, but I think that all the diseases that we see it in the processes, things like head injury, I mean, it's very non-specific, but you see it rise and spike in anything where you get neuronal damage.
Sure. Thank you.
Thank you. We have other question from the chat. Another on riluzole. Are there any difference in neurofilament levels in patients given riluzole in the disease early onset versus when the disease is established in already advanced stages. So is there an effect on the neurofilament concerning the initiation?
I don't think that's been looked at just for sort of numbers, really. There was a study came out a few years ago where they sort of reanalyzed some of the riluzole data and,
Yes.
a suggestion that perhaps its activity wasn't occurring until later in the disease, which I think biologically that's quite hard to understand. I mean, we don't know exactly how riluzole works, but yeah, I think that study hasn't been done. But as I say, I just think we've got to get something better than riluzole, and this is the way to do it.
Well, no one really knows the mode of action of riluzole.
No.
Yes. We have other questions. Another one for neurofilament. So if neurofilament levels correlate strongly with disease activity and progression rate, how does this biomarker correlate with overall functional severity and individual clinical symptoms? And if it does not correlate strongly, how would we objectively measure the effect of the drug in patients' functional status?
Yeah, well, it does correlate as I showed. So that's why it works. It correlates very strongly with disability and survival, and in the individual is predictable over time. So that's why it's so powerful and is unique, really. So yeah, for all those reasons, it's a great candidate.
Another question from me from neurofilament. Is neurofilament for you best biomarker for diagnosis or prognosis? Do you use neurofilament for the diagnosis of ALS or for the prediction of the progression of the disease?
Definitely not for the diagnosis. So the diagnosis of ALS is a clinical diagnosis, and then once we've made that diagnosis, we use neurofilament to stratify, so it's a prognostic marker. We did a study actually that's published in Brain Communications by Jenny Davis, and we took 100 patients referred to the clinic prospectively, and myself and Alex and Kevin, when we saw them clinically, we had to decide if they had ALS, or we had to decide if they had something else, or we had to decide if we weren't sure. There was only 3 outcomes. We had to say one of those 3. And so we did that prospectively, and then after we'd done that, we then looked at their neurofilament levels. And neurofilament wasn't adding anything to our clinical judgment. And there were obviously people with other neurological conditions who had a slightly raised neurofilament. There were people with ALS who had low levels because they had slow ALS, and the people who weren't clear, it took more time because they were slower. So it has no role in making a diagnosis of ALS. I think there's still a study to be done probably in whether it can provide some reassurance for that group of people who many neurologists struggle with, young men who go to the gym and become obsessed with fasciculation. There may be a negative predictive value in I think that would be a good study to do, actually, because they often get very anxious. They're not reassured. They keep sending me videos of their muscles, and it's very hard to get them to relax. So, there might be a value there, but it's very important not to use neurofilament as a way to make a diagnosis of ALS, because it doesn't have that specificity.
Okay, even for the confirmation of the diagnosis, if you have many clinical signs, you are not sure of the diagnosis, and you have a high level of neurofilament, is for you? Or?
I mean, I think I could imagine a situation where I might say to a patient, look, you haven't got very many upper motor neuron signs, but you've got a very high neurofilament level. But I would have already realized if they've got a very high neurofilament level, they'll have been changing quickly. They'll have come after 12 months having been completely well and now pretty disabled. And there's not many other diseases other than ALS that does that. So yeah, I just don't think there's many times I'm using it in that way. It's something we do within a trial setting to stratify patients, and what I've demonstrated here is to look for drug effects.
Okay. the last question from Bill Eldridge, excuse me for the pronunciation. How to support early stage drugs treat NEM patients with appropriate biomarkers?
How to support, sorry?
Early stage drugs treat NEM patients with appropriate biomarkers.
Well, I suppose what might mean there is if people have started to take things already. I mean, we do allow people who are on riluzole to take part in experts, i.e. to add another drug on top of that, because we imagine that if they've started something, their new level of neurofilament, provided they've been on it for at least a month, will be their baseline, whatever. And it's actually one of the attractions is that this platform might be the way that we could start to do combination therapies, because it may well be you need more than one drug. So I think it lends itself well to that, really, because as long as the person is taking a stable dose of whatever they're taking, we can then say, okay, this is your baseline neurofilament. We're adding a new drug in. Let's see if it lowers it.
Okay. Thank you very much, Martin, for the talk and the reply. Thank you to all of you to attend this nice and very fruitful talk. Thank you, Vincenzo, for the organization, and of course, for the organization too. See you soon.
Thanks a lot.
Bye.
Thank you.
Bye,
Thank you
Martin.
very much. Bye.
Bye.